I'm really delighted to be able to share with you today our data from our new hMPV challenge model. So I'll take you through how we developed the virus, how we manufactured it, and then also show you the clinical development pathway of that and the actual clinical data. And now it's ready for vaccine testing. So the hMPV challenge model is actually building on the success of the RSV virus model that we already have. Of course, this is a member of the same virus family. What we've seen with the development of these RSV A vaccines, we used the RSV A model in human viral challenge studies and got really great data to show the efficacy as shown here. So these were the first indications that you can actually vaccinate against this really key pathogen. And there's lots of decades of work gone into this and really struggled to get a good vaccine for RSV A. And these challenge models were the first proof of concept showing actually you can get efficacy against these viruses with these pre-F based vaccines. And these were then really predictive about what might happen in the field and actually a key component of bringing the RSV A vaccines into human population. Of course, they're now saving many lives. So those research groups have now moved on to looking at other pathogens from the same virus family. Of course, hMPV is from the same virus family of RSV A. So we're building along the knowledge of this family when we set about developing the hMPV model. So the first thing when you're developing a model is to have the clinical isolates. We need to have naturally occurring clinical isolates because we want these viruses to be representative of what's actually circulating in the community. So we take clinical isolates from people who have been infected naturally in the community. Then we produce a seed virus, then it goes to GMP manufacture, then we do a pilot phase of the clinical trial, looking at do we think we've got about the right dose, and then once that done that, we then move on to completing the characterization of this virus to give us a full body of clinical data that's needed to power future efficacy testing. So as we've gone about setting up a whole panel of different viruses here, because what we've seen is a trend from once we've got market authorization for the RSV A vaccines, there's been a trend from those researchers to move on now to make combination vaccines. So perhaps in combination of RSV and hMPV, or even sometimes trivalent vaccines with RSV, hMPV, and parainfluenza virus. So at hVIVO, we're trying to make sure we've got challenge models available to test the efficacy of each of those components. And this slide here just shows you the readiness of each of those models. So of course, in purple, we've always got the RSV A Memphis virus that's been available and been very successfully used to date. That's also ready for testing. And now we've got an RSV B model that's ready for testing. And what I'm able to go to you and share more data with you today is our hMPV model, also ready for vaccine testing. In addition to that, we've got a brand new contemporary strain RSV A virus that's been developed and needs now to go through into clinical testing, and we've got clinical isolates, but the PIV, which can then also move forward into seed virus testing and seed virus development and manufacture at a later stage. So we're really well set now to develop this model further and have different viruses available to test for these combination vaccines. So coming back to the purpose of our talk is to talk to you about the hMPV model. So to start with, we really honed in and got good clinical samples, good quality clinical samples. This time they were from pediatric volunteers, pediatric patients, should I say, who actually unfortunately got infected with these viruses in the community and were hospitalized to the point that they were actually in intensive care. So these parents very kindly gave their consent to take these clinical samples to therefore get a virus which we can then develop these models for, with the hope, of course, of bringing through models and therefore test vaccines and antivirals and get better countermeasures to protect against these really important pathogen. These challenge models can be really valuable in testing vaccines and fast-tracking the development of them. So if we can use these models to fast-track the development of vaccines, that is really what we're about at hVIVO, to develop products and bring them to humans faster. So we start off with these clinical samples, a number of different clinical samples from different sources, and then we triage them, and then from those triage, in the triage we're looking at safety, we're also looking at their ability to grow in the actual GMP cell lines, and whether they have potentially any other amphitricious agents in those samples, which of course would eliminate them from further use. So those which grow the best, and we can develop the methods for that, we then go through into GMP manufacture and produce a GMP grade of this virus. This means we're producing essentially a medical grade version of a naturally circulating virus with very minimal passage, so it's had very little chance of having cell adaptation as we move forward. And then a very basic step is then remove any cell debris, isolate the virus, and then aliquot that ready for human use. Very important aspect that these are vials that are ready for inoculations. So they're frozen down in different dilutions. So once we've worked out the titer, we can then pick the dilution that's best for clinical use. And we don't need any further manipulations outside of the GMP suites. This really helps minimize any variabilities. There's no further manipulations. Whole GMP patch is aliquoted and treated in exactly the same way. So then we move on to clinical testing, and we now have 28 persons that have been inoculated with this virus to complete the study. But it was started off with a pilot study phase of 8 individuals and then extended out to another 20 individuals, and I'll show you data from both of those. And both of those phases, the pilot phase and the extension phase, went through the same basic setup, where they come in for initially a screening visit, and that first screening visit is really the very first question we're asking is, what is their antibody status? And we measure their antibodies at that point in time, and we start the collection of the data that looks at their overall health status. It's really vital that these people are actually very healthy. They need to be able to expect it and have a good immune system and expect it to clear the virus naturally. These models are made safe not by selection of the virus, but made careful selection of the volunteers. If we gave these viruses to risk populations like an elderly person or a very young baby, for example, then they could and may well have very severe disease. Of course, we will need these models to be absolutely safe. So very careful selection of volunteers by very extensive safety monitoring and good understanding of their current medical status. That's done at screening, and then they come into the quarantine 2 days prior to inoculation where we recheck their health status. In addition, at that time point, we're also then screening them for any other viruses they may have picked up in the community. So, of course, if they've got any symptoms, they'll be immediately discharged, but they may also have a subclinical infection. So we take a swab at that point in time, and then that is gone for diagnostic PCR testing. So even at the subclinical level, we're testing to make sure they haven't got any other pre-existing respiratory viruses that could confound the data later on or even confound safety. So anyone positive at that stage will then be discharged and not inoculated with the virus. So having established safety and having established they have no other respiratory infections, we inoculate them on day 0, and then we follow the disease trajectory throughout the entire time course. So for each day, we collect symptom diary cards and also do nasal swabs to look for the viral load and measure the quantifiable how much virus they're actually shedding at the time. So this goes into a little bit more detail now about how we measure that disease. So I mentioned the symptom diary card. It's actually a categorical symptom diary card, meaning we ask the volunteers to score on 0, 1, 2 or 3, depending on severity of the symptom that they have. So for example, if they had just a little bit of sneezing, they may score a grade 1. If they felt it was really quite bothersome, they may score grade 2. If it's stopping them from doing their daily activities, only then really do they score a grade 3. So it's quite unusual to have grade 3 symptom scores, because that really would be stopping them from doing their daily activities. And most respiratory common cold viruses, of course, don't do that level of severity. So most of the symptoms we expect to see for infected individuals will be grade 1 and grade 2. Now, we ask them to do that for the full length, the full list of the different symptoms that they could potentially have. In addition to that, we ask them to fill out a patient perception questionnaire on whether they perceive they have a cold or not. So, very simple question. Do you perceive you have a cold? And if the answer is yes, is it worse or better than yesterday? And this can be really useful as a trigger for dosing of an antiviral. So instead of triggering on PCR-based products, you can trigger on a question of when they have a perceived when they have a perceived cold. And of course, that's really key, because that actually translates really well to how they might be recruited onto a field trial. So always with challenge models, we're trying to make sure that they do translate and predict what's going to happen in a field trial for those products later on. Now, that's exactly what would happen. Of course, you have to have perceivable symptoms and feel quite enough bothersome that you pick up the phone to your researcher that you're recruited on the clinical trial, or go into the physician. So to do that, you need bothersome symptoms. And this grade 2 really translates really well with that, as well does the patient perception questionnaire. All of the respiratory virus models that we have, of course, cause running noses. Therefore, we need to collect and understand how much mucus discharge that they have. And then we do this by having a crude measure of measuring the total discharge into the tissues. So we have pre-weighed tissues given to the volunteers. If they need to blow their nose, then they blow their nose into these pre-weighed tissues. They go into a sealed bag, and then we have a total weight for the day. So we can have, by that measure, we can have a weight of nasal discharge. A very crude but very effective measure, actually, of what's going on with the clinical symptoms and disease. Now, what we have here for you is the pilot data. So each individual subject is represented by a different curve. So this is the very first look at this brand new hMPV challenge model. And what we can see is in the line is the PCR curve, and in the green bars is their symptoms. So just as you would expect for a respiratory model, we see a good range of disease severity and a good range of symptoms. So on the top left, we can see very robust PCR levels and very high symptoms indeed. In fact, we've got symptoms in the majority of subjects, and we've got PCR positivity in nearly everybody. So everybody has some level of PCR positivity. Not the bottom right would not have met our infected definition, but one of the time points did have PCR positivity. So really high infection rates and good, robust disease curves. This pilot data gave us great confidence that actually this is working very nicely and was the right concentration to move forward with. So we're able to move forward then into look at the rest of the model, where we then inoculated further 20 volunteers to get a bigger body of data. Now, we all know with respiratory viruses you can get asymptomatic infections, and that's exactly what's been depicted here. So we've got individuals with PCR curves but no symptoms. This is exactly what you'd expect, and of course is what happens naturally in the community. We all have sometimes asymptomatic infection as well as sometimes a symptomatic infection. So in a challenge model, when you're giving this to all comers, you expect to see this full breadth of disease, and that is indeed what we saw with the pilot study and gave us great confidence we're onto the right thing, and therefore moved on with the rest of the study. And we summarise now the rest of the study, which is a total of 28 volunteers, and we can see we've got really robust disease. And when you're looking at endpoints for these trials, particularly for vaccine endpoints, incidence endpoints are often a key endpoint. So we can look at different measures of incidence. For example, PCR positivity. Are they positive by PCR from their swabs, which are taken twice daily? That's the most basic measure of infectivity, and see really robust levels here with this hMPV. And of course, then we'd start to factor in symptomatic disease as well. So how many symptoms do you have? Do you have any symptoms at all? And actually what we find is the most used primary endpoint for all of these studies is moderate to severe symptomatic infection. To meet this definition, they not only have to have PCR positivity and meta-infection definition by PCR in that way, but also they need to have symptoms of a grade 2 or higher. So those moderate severe symptoms that really would be quite bothersome for them, that I mentioned earlier, really translate well to field trials. And this is what we see is on this model is a really robust level of that at around 50% of those moderate severe infections. And on the right-hand side, you can see how the time course of this disease progresses over time. So we got in the blue bars there, in the blue line there is the PCR, and in the green is the actual symptoms. You can see a very nice correlation of the timing of the peak symptoms and the timing of the peak PCR. So when you set up a new model, it's very hard to know what a good model looks like. So actually, we're in the good fortunate position here that we've had the RSV A model has been so extensively used and already proven to translate very nicely into what is able to be predicted to be a good, useful vaccine, that actually that sets the benchmark here for a member of the same virus family. So that's on the right-hand side. And in this, what we're showing here is the data from all the placebo subjects that we have in our database for the prior studies of the RSV A, hence the very small error bars, because it's a big, wide data set. And we can see if we compare the disease severity then to hMPV, which is on the left-hand side, you can see they compare very favourably indeed. So very similar magnitudes of disease from the lower levels right up to peak, very similar magnitudes, and actually very similar duration of the disease. What we can see in hMPV, it does peak a little bit earlier, so it's a bit more of acute disease. So you're peaking a day or so earlier than we did in RSV, but actually the levels of disease that we see are very similar, and it does take a similar amount of time to clear the disease, about 12 days. So this gives us a lot of confidence actually, now that we know that the hMPV model is giving very similar levels of disease to what we see with the RSV A model, gives us huge confidence that this is going to be a really useful model moving forward for hMPV vaccine testing. So if we look in a bit more detail about what sort of symptoms we're seeing, we can see in the purple are those grade 3, In the yellow are grade 2, and in the green are the grade 1 symptoms, so the lowest level symptoms. As I mentioned earlier, the grade 3 symptoms are very unusual to see in human viral challenge models, because you're asking them, do they have symptoms that are severe enough that they would stop them doing their daily activities? And that's unusual for a cold symptom to stop you doing your daily activities. You may feel quite poorly, but they rarely stop you doing your daily activities. So therefore, we don't see very many grade 3 symptoms typically. What we see here for the hMPV is low levels of grade 3, but actually we are seeing really robust levels of grade 2, which is that key moderate to severe endpoint that I mentioned earlier. In addition, we're seeing good levels of the grade 1. So not surprisingly, it's mainly an upper respiratory tract infection, as depicted by the symptoms that we see, with also good involvement of systemic. And actually the only indication of any lower respiratory tract involvement at all is small measures of some cough in some individuals. So when we set up this model, actually we've set it up inoculating the volunteers throughout the whole breadth of their antibody status. So we've always been asked, can we set up a human viral challenge model that actually represents more what would happen in a field trial? So in field trials, of course, you don't know the immune status of those volunteers. So in this case, we do know the immune status. We measured their antibody levels prior to inoculation with the virus, but we chose to not exclude any levels when they went into the study to see, can we get robust infections without pre-excluding people who have high levels of antibodies, because then if we were able to do that, we're able to have a model which can better represent what's going on forward. And that is indeed what we saw. We've now produced a model where we haven't pre-excluded people with high levels of antibodies, but we're still seeing robust levels of disease. But of course, it's always important to understand what impact do those antibodies have. And on this slide, we can see the startings of that analysis. So we're asking the question on this slide, so are we seeing any responses to inoculation? So we measure the baseline antibodies, and then we measure the antibodies after approximately 4 weeks after they've been inoculated with the virus. And you can see in over 90% of the volunteers, we're actually seeing an increase in antibodies. So a response and expansion of the antibodies in response to that virus infection, just as you would expect to see with a high infection rate that we have in this model. And we also see that a good level of those had higher than twofold changes from the baseline as well. And if we look at how that correlates, we can see actually there really is a correlation between the antibody levels at baseline and severity of disease. So on the left-hand side, you've got AUC, so area under the curve, which is a measure of the total amount of disease that they see by PCR, and see there's a good correlation between AUC and the baseline levels of antibodies. And similarly with peak virus, there's also a good correlation. So although we set this model up with the idea of seeing if we can actually see infections without pre-excluding, which we have been able to do, this data tells you that you may be able to increase the level of severity seen on the majority of volunteers even higher if we did pre-select. So this model is ideally situated to be able to do either, whichever you would prefer. So if we want to keep the N number very, very low, we can pre-screen the volunteers, exclude those with the highest levels of antibodies, and therefore maximize the number of people we see with higher levels of symptoms, minimizing the N number and making the study very efficient. If you want a study which is more real world, we can not exclude anybody, but stratify to make sure there's no random bias. And in that case, we have a more real world answer about what's more better translates to what happens in a field trial. So in summary, we've got a great hMPV model here with a really robust disease, and we've got good AUCs, good incidence of the higher levels of moderate severe infection, which of course is really the key endpoint that's used for vaccine efficacy testing. We've also demonstrated by the antibody level measurements that we've done that you can actually get this level of robust disease by putting people into the trial without pre-selection of their antibody status. However, we have shown a good correlation of the pre-existing prior to inoculation antibody levels with the levels of severity. So if the aim of the trial is to maximize the people with that moderate severe infection, and therefore minimize the size of the study and have a very efficient study, then we can do that by pre-selection of the antibody levels of those volunteers that they go in. However, if the aim is to have a model that translates really very nicely to field trials where you don't pre-select individuals, this model can run like that too. So really got both options available for this fantastic new model. Thank you for your attention.